everybody, it's Miss PG from your WCA Biology class, and we are doing a video tutorial today for Unit 5, Lesson 10, Gene Expression. And so for this one, ladies and gentlemen, this is going to be a little bit of a review from previous units, or previous lessons, I should say. Some of these terms we've all kind of seen before. There's a couple of new ones, so that's why I decided to do a video on this one, but a lot of it is kind of review from what we had done in kind of a couple of previous videos, which is a good thing because some of the stuff's kind of hard, so it's nice to be able to see it come back and kind of re-emphasizing it so that you guys don't forget and maybe get a better understanding of it the second time through. Just as a reminder, as always, there is a notes guide that kind of goes along with this. Remember that you can find this in the link underneath the video on the biology website. So that's available to you to use. You can just have it open and you can split your screen kind of like I'm doing here, so you can kind of watch the video on one half and take notes on the other half, and that allows you to be able to kind of keep up with what I'm talking about here today. So today I decided it was probably best use of my time and yours to actually kind of go through some of the stuff in the lesson, simply because, like I said, it's a little bit of a review, so it kind of helps you to process and understand what's going on in the lesson. So going to slide 2, kind of goes on and talks about what it means to be genetically identical. So that's kind of our first term here. Genetically identical daughter cells. So as we can see here, this little illustration, they have it in the lesson. I put it in my whiteboard as well. The process of mitosis is what we're talking about. So mitosis is cell division that ends up yielding genetically identical daughter cells. Starts with the parent cell, which is what we talk about here. And in this example, we have a cell with 3 chromosomes, simply because it's just easier for us to be able to draw it, to model it, and to kind of see what's going on with just a few chromosomes than a human cell, which has 46 chromosomes or 23 pairs. It becomes a little tricky to be able to kind of keep track of everything. So again, remember, first, before cell division even happens in the cell cycle, this is the S phase of the cell cycle, the synthesis phase. DNA actually has to replicate in that phase. That's one of 3 phases in interphase. So interphase is that part of the cell cycle where it's not going through mitosis at that time, but it is just doing other things. So it's either, well, remember there's the G0 phase, which is where cells kind of go when they're not doing anything. They're not getting, well, they're doing what they need to do, but they're not actually getting ready to divide or dividing at that point. And then once they go through that G0 phase and they finally just need to divide, some signal to the cell determines that division is going to happen pretty soon. It first has to grow and get bigger and has to replicate all of the organelles in the cell. And then that's the G1 phase, and then the S phase. And the S phase is where the chromosomes get copied. And then they get copied because they're going to divide, and we want each daughter cell to be exactly the same as each parent cell. So once it goes through that process of mitosis, right, those sister chromatids will separate here, half going to either side of the new cell, or the larger cell isn't divided yet at this point. And then the 2 new cells that form each have 3 chromosomes, just like the original did. So these 2 cells are genetically identical to each other and genetically identical to the original. And as time goes on, these cells will replicate eventually, and they'll create genetically identical cells. And so that means that, every generation essentially of replication yields the same cells that you started with. That's the idea behind mitosis. Mitosis gives us genetically identical cells. For example, when I have my skin, I don't want to have like a bunch of different cells in there. I want them all to be the same. I want to have like a nice even layer of skin, right? So the cells are all genetically identical to each other. Then we have another definition here, and that's called the definition is proliferation. And what that really just means is to increase in number. So in this case, we're talking about cells by replication. We're increasing the number of cells by replication. Proliferation can really refer to anything that's increasing in number, but in this case, we're talking about cells by replication. And then it says, after mitosis, a cell that is not specialized will undergo gene expression to become specialized. So cells start off as kind of these generic stem cells, and then they need to be specialized so that they can perform whatever function they're destined to perform, right? So cells that are destined to be able to secrete acid in your stomach are able to do that, because every cell in your body is going to be different cells that are in different types of body tissues have different functions. And so to be able to be specialized to perform that function, they need to undergo something called differentiation. So then it talks about stem cells here again, and we've gone through this a couple of times already. But that's just a reminder that there's 3 types of stem cells, that we have 2 types that are specifically found in embryos, the totipotent and pluripotent stem cells, and that these have kind of more of an ability to differentiate into more things. They're more kind of plastic-like in that they can become kind of anything moldable, but we only find them in embryonic stem cells. And then we have multipotent stem cells, which are found in adult stem cells or a developed organism. And these are already differentiated a little bit, so they have a limited ability to become other things. So you've got different types of multipotent stem cells in your body. You've got skin cells that are multipotent, and they can turn into different kinds of skin cells. You've got bone cells that are multipotent, and they can turn into different kinds of bone cells, but they're just limited in that they can only kind of become a specific type because they already are somewhat differentiated. All right, so again, that stuff's kind of review for you as well, right? So then we go into what gene expression is, because remember, in order for a cell to become differentiated, it has to undergo gene expression. So gene expression is, again, stuff we've talked about before. This is the process of transcription, translation, and folding. So this is really the process of protein synthesis, which we talked about a couple of times. He spent a whole lesson talking about it as well. And just recalling that transcription takes place in the nucleus, and this is the part where the DNA is unzipped, and then part of the DNA, like one side of it, is the template from which messenger RNA is made. That part is called transcription. Messenger RNA then moves out of the nucleus through one of the little nuclear pores in the nucleus, and it finds a ribosome to attach to. This is where the second step of gene expression occurs, called translation. And translation is where that messenger RNA is read it's read 3 base pairs at a time, or 3 nitrogenous bases at a time. And we call that 3 base sequence a codon. So we say it's read 1 codon at a time, which is basically made up of 3 bases. It is read by, it moves through the ribosome, and these transfer, or tRNA, bring down little amino acids that match up with it, right? And then they assemble 1 amino acid at a time to form kind of a chain of amino acids all linked together. That long chain of amino acids is called polypeptide chain, and it is not completely a protein yet. So we've kind of referred to this before, but I haven't quite elaborated on it. So our third kind of stage of gene expression is folding. So the final step of gene expression is folding the proteins into complex ribbons and strands. This step is important because without folding, the proteins would not be useful in the body. The folded proteins that the ribosomes make determine a cell's function. So I have kind of alluded to the fact that proteins need to be folded. And so that is the final stage of gene expression. Even at this point, the protein may not be completely functional, because remember, the protein then gets shipped off to the Golgi apparatus, and there oftentimes different lipids or Other things are kind of attached to it, different sugars and stuff like that, to complete the protein. So sometimes the proteins need to have stuff attached to them in order to be completed. It goes on to say a cell may be able to replicate itself during differentiation, but not all differentiated cells are able to replicate or proliferate often. It comes to a point where if a cell is no longer able to proliferate or to replicate, we say the cell is in a state of senescence. And we've referred to this word before as well. So senescence means that cellular reproduction is going to stop. And sometimes this is permanent, sometimes it's temporary. When it becomes completely permanent, where it cannot do it anymore, no longer can go back to active replication, then we say it has undergone terminal differentiation. So senescence may just be kind of a holding pattern where it's not currently dividing, but it could potentially do it in the future. But once it's no longer able to anymore, we say it's undergone terminal differentiation. Okay, and so that is kind of an overview of this lesson. Again, lots of review here, but I thought it was worthwhile to go through again because some of this stuff, like I said, is a bit tricky, so it's nice to hear it twice. So if you have any questions, let me know. Otherwise, that's the end of this one, ladies and gentlemen, and I will see you in the next.